All right, today we're diving into one of the most incredible little pieces of machinery inside your body. We're going to look at what is essentially a biological post office, a master logistics center that keeps your cells running like clockwork. Let's get into it.
So, let me just start by asking you a question. What do you think is the most sophisticated and reliable shipping center in the entire universe? Some massive automated warehouse, maybe? A global logistics network? Well, the answer is actually way smaller.
In fact, it's microscopic. We're talking about the Golgi Apparatus. You might also hear it called the Golgi Complex or Golgi Body. And here's the thing, you don't just have one, you have trillions of them, working non-stop 24/7 inside almost every single cell in your body.
To really get what the Golgi does, the best way to think about it is like a combination of a post office and a factory assembly line. It takes in raw materials, it processes and packages them, and then it ships them out to exactly where they need to go. It's the grand central station of your cells.
This whole complex job really breaks down into four main steps. First, it receives unfinished proteins and lipids from its neighbor, the endoplasmic reticulum, or ER for short. Then it modifies them, tweaking them, often by sticking on these complex sugar chains. Third, and this is absolutely crucial, it sorts everything by slapping on a molecular shipping label. And finally, it ships them out, packaging the finished goods into these little transport bubbles called vesicles and sending them on their way.
So if the Golgi is the cell's post office, what happens when a package gets sent to the wrong address? Well, inside a cell, a single delivery error isn't just an inconvenience. It can be a complete catastrophe, which really shows you just how vital the Golgi's accuracy is.
You see, one of the Golgi's most high-stakes tasks is handling the cell's digestive enzymes. These are incredibly powerful chemicals designed to break down waste inside a special compartment called a lysosome. Think of the lysosome as the cell's recycling center, or maybe its stomach. You definitely do not want those enzymes just floating around loose.
And the whole thing hinges on that shipping label. On the left, you see a healthy Golgi doing its job. The digestive enzymes get tagged with a specific label, mannose-6-phosphate. Receptors spot that tag and make sure the enzymes get routed correctly. But look at the right. If the Golgi messes up and doesn't apply that tag, the whole system just falls apart. The enzymes are accidentally shipped outside the cell, leaving the lysosome totally empty and useless.
And this is not just some textbook theory. No, this exact sorting error is the real-world cause of a devastating group of genetic conditions called lysosomal storage diseases. Without those enzymes, the lysosomes can't break down waste, so the waste just builds up and up to toxic levels, causing all kinds of horrible damage. It all comes down to one tiny mistake in the cellular post office.
Now here's where it gets interesting. For decades, scientists knew what the Golgi did, but they were in this huge debate about how it actually worked. It was a genuine scientific whodunit, and it all centered on a pretty simple question about how things move through the system.
Let's go back to our analogy. Picture a package moving through a post office. Does the package get put into a little cart and wheeled from one fixed sorting station to the next? Or does the entire sorting station itself actually move along a track, kind of like a conveyor belt, carrying the package with it?
Well, these two ideas were the basis for the two main scientific models. The stable stations model said that the Golgi's flattened sacks, called cisternae, are fixed in place. Cargo just hops between them inside tiny vesicles. But the moving platforms model argued that the cisternae themselves are the transport. They form at one end, mature, and physically chug along through the whole stack.
So, who was right? How did they solve it? Well, the key piece of evidence, the real smoking gun, came from looking at really big cargo. Molecules like procollagen, which is basically the scaffolding for our body's tissues, are absolutely enormous. And scientists realized there was no way they could be stuffed into those tiny little vesicles. This was a huge problem for the stable stations theory. That one little detail about the big cargo completely tipped the scales, and it pushed scientists toward a final answer. So, what was the verdict?
The winning theory is called cisternal maturation, which is really just a fancy scientific name for our moving platforms model. The idea is that the Golgi is this dynamic, moving conveyor belt. A new compartment forms on the receiving side. It physically moves forward while changing its own enzymes. It literally matures as it goes. Then when it reaches the shipping side, it breaks apart into all those vesicles. And this just beautifully solves the procollagen problem. The big cargo never has to squeeze into a tiny vesicle. It just hangs out and rides along inside its moving platform, the cisterna, all the way from start to finish. It's such a simple and elegant solution.
And pretty much the entire scientific community agrees. As this quote from a major review on the topic puts it, cisternal maturation is the best explanation we have for how this works across almost all complex life, from fungi to plants to you and me. But hold on, the story isn't over. Because the Golgi isn't just a post office, it's also a ridiculously sophisticated factory. And if you thought the shipping part was complex, well, this next bit of molecular choreography is going to blow your mind.
In plants, the Golgi has an even bigger manufacturing job. It's responsible for building the complex carbohydrates, the polysaccharides, that make up the plant cell wall. You know, the stuff that makes plants strong and sturdy.
Okay, to really see how amazing this is, let's zoom in on just one specific thing it builds, a polysaccharide called xyloglucan. It's got this long sugar backbone with all these complex side chains branching off. Think of it like a custom-built, super specialized piece of rebar for the plant. So, how many different specialized enzymes or molecular workers do you think it takes to build just this one molecule? Any guesses? The answer is 7. 7 different workers. But here's the truly incredible part. These 7 workers don't just work one after another in different parts of the Golgi. Nope. Evidence shows that they actually clamp together to form a single, giant molecular machine, a multi-protein complex that works in perfect sync.
This is the perfect mashup of everything we've been talking about. You've got a team of 7 specialist robots, all locked together into one assembly unit, all working on a product as it glides past them on the cystolar maturation conveyor belt. The level of coordination and efficiency is it's breathtaking.
From sorting mail, where one mistake can mean life or death, to running these unbelievably complex assembly lines on a moving platform, the Golgi is a true master of molecular choreography. It's just a powerful reminder that even after all this time, the tiny world inside our own cells is still hiding some really profound secrets, just waiting for us to figure them out.